Good morning. I'll take the prerogative of being up on the stage and take off my mask. Thank you. It is such a pleasure to be here. What an exciting time. I feel we've been in some kind of a bubble that we haven't been able to see each other for the past couple of years. Many familiar faces, and we're here together at this magnificent building and institute to really celebrate an incredible day. And that's the keynote lecture that's for the prize that's being awarded to Professor Robert Zatorre. So let me tell you a little bit more about why we're here today. Just to remind you, my name is Karen Abraham. I am the scientific director of the Scientific Committee for the Fondation Pour, audition. This is the body that provides grants and other type of funding, which I'll tell you about in a few minutes. And I'm also the Vice Dean at the Faculty of Medicine at Tel Aviv University. So I transitioned the other day from literally summer to winter, but really exciting to be here. And after not being here for 2 years, I'm reminded really what a spectacular and beautiful city this is. You're very lucky for those of you who live here. So we are here today to really award this prize. We're just so proud of Professor Robert J. Zatorre, who's the laureate of 2021 Scientific Grand Prize of the Fondation Pour, audition. And we welcome you and your family who's arrived for this really wonderful day. I'm going to give the opening, and then the closing will be done by my colleague, Professor David Moore, who I think many of you know is a co-member of the Scientific Committee of the Fondation Pour, audition, and he arrives from Cincinnati, another cold place. Okay, so if you'll allow me, I would like to spend just a few minutes telling you about the Fondation Pour, audition and the activities that we do. Okay, so first, and very exciting, this is France's first public interest foundation that focuses on hearing health. And for many of you, some of you who come from places around the world, I think you know that this is truly a unique place. And so while it's France's first, I think it's a first in many cases in the world. It was created by Françoise Bettencourt-Meyers and Jean-Pierre Myers and the Foundation Bettencourt-Schuller. And it's been recognized as a public interest organization since 2015, and that's about how long I've been involved and been extremely fortunate to be part of this really incredible endeavor. There's a threefold mission that the FPA has. It's to accelerate research and innovation, it's to improve the everyday life of people with hearing impairment, and to raise awareness about hearing health, not only in France, but the rest of the world. We have an extensive research program, and I'm extremely fortunate that I have these partners in crime here. And these are my fellow members, Dr. Bastian Epp, who arrives from Denmark. You want to give a wave there in the back? Dr. Frank Lin, who's at Johns Hopkins University, who is going to be Zooming with us during this week from Baltimore, but wasn't able to come here personally. Professor Larry Lustig, who, give a wave, is over in the third row. Professor Brigitte Malgrange. Brigitte, where are you? There you are. Professor David Moore, who I already introduced. Professor Cecile Perietti-Winkler, who's in the second row, and we're very excited that you've recently joined us, one of our newest members. Dr. Jonathan Peel, who's at Washington University in St. Louis, also will be Zooming with us. He won't be here present physically. Dr. Gabrielle Saunders, who's from the University of Manchester, and another one of our new members, very lucky to have her, and Professor Terry Van den Abel, who is from the Robert Debré University Hospital in France, and there he is, also a new member. So we've had a bit of a changeover, and this is really good to keep things going and keep things fresh, but we have the advantage of our more senior members in the group, so we've been able to keep the historical perspective, even though it's a short history, going, I think, very well. And I'm so lucky this group, we really work tirelessly round the clock to get all of this work done. Since 2013, the support has been to cover 74 laureates at a level of 14 million euros, so really quite an extensive amount. We have funded laboratory research, translational research, scientific prizes, one of which we'll hear about today. We've had open requests for funded projects. We very much encourage training and helping promote the growth of the next generation that's going to be very active and proficient in the hearing field. Fellowships for postdocs, for studentships, master's, PhD, as well as travel awards to the Association for Research in Adult Laryngology conference that takes place usually every year between the East and the West Coast in the United States. Now, our support strategy is listed here as to train future French talents to research. It's not just about us. It's so critical that we make sure that we move forward into the future, that we promote French scientific and clinical research excellence, and we discover solutions for the hearing impaired. And I should add, in the second part, we very much also encourage international collaborations, and you can be, coming from outside France, also part of the FPA. So we have been involved in training future French talents, whether it's MD residents, speech therapists, audiologists, ENT residents, and young clinicians. And we really put a lot of an emphasis on trying to get integration between basic research and clinical and translational research as much as we can. We have a large number of research projects that we fund for laboratories, as well as starting grants for the Institut de l'Audition, this really magnificent building that we're sitting in today. And we work on discovering solutions for the hearing impaired. About 2 years ago, we started a call for translational research with the RNID in the UK. Yesterday, we had our grant proposal meeting, and it's been really a great source of pride that together with the RNID, we're able to fund translational research grants. We have the Scientific Prize, which we'll hear about today, and then support for the Clinical Research Network in Audiology, which is also a major priority. So in terms of the research proposals, I think many of you are familiar with them. We both fund basic research, medical and audiological research, applied research, and technological innovation, as well as psychology and sociology. And we've been very fortunate to have really a broad spectrum of research proposals that have been either proposed and or funded over the years, and we're going to see a lot of the fruits of this in a symposium that we have coming up this week. In February 22, we will be launching again another round of calls. This is again the translational research grant with the RNID. Really very prominent funding. Researchers can come from any university, from any country, as long as they fit the criteria that are needed. And again, the emphasis is translational research. We're trying to do as much as we can to ensure that the fruits of the research that is part of the Fondation Pour, audition is going to be applicable for individuals worldwide. We have postdoc fellowships and MD fellowships. We have a call every year, and really we rely on you to help us disseminate this information as much as we can, particularly the clinical side. We find that they don't hear as much about what we have to offer, and I think the more we can spread the word, the more candidates we'll have for these really incredible opportunities. We'll have PhD studentships again and master studentships, so there's a constant rolling of these calls coming out for eligible candidates. We also have the ARO Travel Awards, again to encourage French ENT residents to visit the United States, to take part of this very important conference that occurs every year. This year it'll be in a hybrid mode, both in person and online. The Scientific Prize, again, which is the reason that we're here. And today we're going to talk about the Scientific Grand Prize, which is a very generous 100,000 euros that's provided to an outstanding researcher or clinician in the field. And so who is eligible for this prize? So it's awarded to an investigator in recognition of his or her work in hearing sciences throughout his or her career. So Robert, you can know what this is for. Major discovery in hearing sciences. It is open internationally, and there's very strict evaluation criteria. We have a very distinguished, anonymous, yet prominent ad hoc committee that goes over the applications and nominates one of the winners. That goes to the scientific committee of the FPA for approval, and finally the board at the foundation. And so this is what we're celebrating today. Professor Zatorre, we're so excited that you're here. Professor Zatorre is at McGill University. He's been there for about, I think, 30 years now. And I was born in Canada, so I'm very proud that we share something in common there. And Professor Zatorre is a cognitive neuroscientist, and you'll hear much more about his work. He's going to tell us about speech and music and how he integrates that in order to be able to understand very complex use of sound. So I think the prize that you've been awarded is a very distinguished one, but the auditory cognitive neuroscience that you're talking about is quite unique, and we're very excited to hear about what you're going to tell us about your project. Professor Zatorre has won a number of very distinguished prizes, so we're very proud to continue this prominent trend of yours. Most recently, it was the Heineken Prize from the Netherlands. So, Professor Zatorre, if I can invite you now to give your keynote lecture.
Thank you very much, Dr. Abraham, for the introduction. Merci à tous et à toutes pour être ici. Je suis vraiment très reconnaissant. C'est un grand honneur d'être ici parmi vous. Et c'est un grand plaisir d'être enfin de retour en France après 2 ans. I've really missed Europe in general and France in particular, so let's hope that things go well so that we can have more exchanges and more interactions. And I just wanted to sort of underline something that Karen mentioned, which is that my award is in recognition of the work I've done, but I feel like I'm representing auditory cognitive neuroscience in general. And I love the fact that at this incredible institute, there's a wide variety of research going on in the hearing sciences. But I believe that the previous awards have been primarily aimed at either more clinical work or more sort of fundamental work in the periphery of the hearing system. And so what excites me is that there's the recognition of the entire spectrum where we're going from the periphery all the way to the highest aspects of cognition. And so that's what I'm going to try to talk to you about today. So a lot of my work over the past 30 or so years has been dedicated to trying to understand some of the most sort of higher order aspects of human auditory cognition. And in that context, I wanted to introduce my talk using your motto, which I think is fabulous, à l'écoute de la vie. It's perfect. And so when we think of that, you know, well, c'est quoi la vie auditive? Well, I think it depends on your particular sound environment. If you're a bat, then your sound world consists of extremely high frequency sounds that you would use for echolocation. And it's a very specialized system, and there's some magnificent research on the bat auditory system. If you're a bird, then you would have a different set of neural specializations that would allow you to sing and communicate with your conspecifics. What if you're a human? Well, most people would say, well, this is our communicative system, right? That's what I'm doing right now, doing a lot of talking. And no doubt, that's a very important part of human audition, essential. And there's no doubt that there are important specializations. But I would argue that we have two communicative systems, not only speech, but also music. And I think these are our species-specific communication signals that our brains are particularly specialized for, just like the bat brain is specialized for its sounds, or the bird brain. And so that's what I'm going to try to understand, is the nature of these specializations. Now, I think most people would agree that the speech is a defining characteristic of the human species, but I would argue also that music is similarly a defining characteristic. And if we look at the archaeology, we see, as has been discovered by archaeologists in Germany, these very ancient flutes that were created by Paleolithic humans around 35,000, 40,000 years ago. And what this means is that if people at this time were making instruments to generate music, music must antedate that by a very long time, because you can make music obviously by singing or by clapping your feet or clapping your hands or stomping your feet. So music seems to be very ancient and probably just as ancient as speech. So it behooves us, if we're going to understand the nature of human audition, to understand both of these systems. And historically, music has been kind of a curiosity in some sense. Darwin famously had this to say about music. He says that neither the enjoyment nor the capacity of producing musical notes are faculties of any direct use to man, and so they're ranked among the most mysterious with which he is endowed. So he was sort of putting his hands up and saying, well, who knows why it's there. That's maybe his most famous quote about music in The Descent of Man. But 10 years later, in his autobiography, he wrote the following very insightful statement. He said, "If I had to live my life again, I would have made a rule to read some poetry and listen to some music at least once every week, for perhaps the parts of my brain now atrophied would have been kept active through use." It's a very interesting statement. And then he goes on to say, "The loss of these tastes," meaning these aesthetic appreciations, "is a loss of happiness and may be injurious to the intellect and to the moral character by enfeebling the emotional part of our nature." And I think that's actually the key idea here, is that music allows us to communicate emotions. So does speech, of course. We can obviously communicate a lot via speech, but music is particularly interesting in that respect. So what are these specializations that we're talking about? I believe there are many different components to the neural networks that support our capacity for musicality. But today I'm going to really focus on the sort of input to that system, which is the auditory cortex, which consists of a concentric organization similar to other sensory systems, and which communicates with vast areas of the rest of the brain, in particular the dorsal and ventral streams that you see here, including parietal cortex and premotor regions and frontal regions. I really will focus only on the sort of more basic parts of this system. And I'm going to give you a little bit of historical overview here. So if we go back to our very first neuroimaging experiments, way back in the 1990s, when we were first developing PET scans, I don't know if you can see that image, but we did some very, very simple experiments contrasting the patterns of brain activity associated with hearing speech sounds using some acoustical controls particular to speech, and also the same thing with melodic sounds. And what we kept seeing consistently was this larger left and right response in auditory areas, auditory cortical areas associated with speech, and typically larger right than left response for melodies, even though in both cases there's bilateral activation. Now, this led to kind of, I would say, a very oversimplified view about music and speech being differentially lateralized. And I say unsatisfactory because it doesn't really lead us to understand what is the nature of this specialization, nor what is the mechanism behind it. So we thought, okay, let's try to take apart the different components that might be relevant for the processing of music. And so we did that in the earliest days by looking at patients with brain lesions, and subsequently went back to neuroimaging, as I'll show you. And here's one of the very first experiments that we did with my former student, Séverine Samson, who is here from Lille. And what we were looking at here were patients who had excisions of the temporal neocortex that included the superior temporal gyrus, and also patients with lesions in the frontal cortex. And this is a very simple task that we borrowed from Diana Deutsch, where you hear a target tone. It's then followed by these interfering sounds, and then you have a comparison tone. And your job is to determine if that tone is the same as that tone. And this task is relatively difficult when the interference tones are placed in between, but not so much if there's silence. And what we found was that the control participants were doing pretty well here. Their error rate was very low. The patients who had lesions on the left had a marginally elevated error rate, but really the ones with lesions on the right were much worse off, and particularly when there was an interfering tone. So this already was one of the first hints that we had that maybe auditory working memory depends upon some of these right lateralized cortical structures. We went on to do a bit more in terms of like psychophysical sorts of experiments here with Ingrid Johnsrud and Virginia Penhune, who's also sitting in the audience, and she's my spouse. We've done much work together over the years. And in this case, Virginia developed a method to identify the size and location of the excision in those sorts of patients. And this allowed us to divide the patients into those whose lesion extended into the lateral portion of Heschel's gyrus, which is the gyrus that contains primary auditory cortex. And we gave people this very simple task where they had to determine if two tones were rising or falling. And this is a staircase psychophysical procedure, so we look at the threshold. And the main finding here is that the patients whose lesion is on the right and whose excision encroached onto this lateral Heschel's gyrus area had a vastly worse threshold for determining the direction of pitch change, contrasting markedly with the patients with similar lesions on the left, who, if anything, were actually slightly better than the control participants. So here's a very dramatic difference between right and left. Importantly, these subjects were able to do the task. They just needed a much larger difference in frequencies between the tones in order to perform it. So it's not that they could not do the task, it's just that they were not really able to perceive the very fine-grained differences. So the interim conclusion from these earlier studies is that there seems to be some kind of specialization for certain aspects of the processing of pitch patterns, but we obviously wanted to go further than that, and in particular we wanted to follow up on this idea that maybe one of the important characteristics of the right auditory cortical system is its ability to resolve fine frequency differences. And so in various experiments we've found evidence that supports this idea. This is a study done by my late colleague Krista Hyde, whom we lost last year tragically. And Krista did a very nice systematic study where we presented a pattern of tones and we systematically varied the size of the excursion in this pattern. And this was a functional MRI study. And what we looked for were brain areas whose activity scales with the increase in this pitch pattern. And what we see is that there's a significant response in this right lateral region, which roughly follows a quasi-linear response, more activity associated with larger frequency differences. And we subsequently replicated this with a slightly different paradigm using these sort of micromelodies that we call. I'll tell you more about them in a second. This was an experiment that we did with Karine Delamot and other French postdocs. We have a lot of French people in Montreal. And what we found here was that, again, the response on the right side tended to be more steep than on the left, as you can see in these graphs here. Using, again, these micromelodies, another of my former students, Reiko Matsushita, looked at whether we could get causal evidence for the importance of the right auditory cortex. We know from those lesion studies that if you lesion that area, there are certain difficulties, but we wanted to do this in a systematic way with healthy participants. And we can explore this using brain stimulation techniques. In this case, we use transcranial direct current stimulation. And with the help of our colleague Sylvain Bayet, another Frenchman, we were able to figure out how to collect MEG data, magnetoencephalography, with the stimulation electrode in place. And in this paradigm, we're taking these micro melodies. These are, again, melodies that fit within the cracks of the piano, if you will. So the difference in pitch between two adjacent tones is called 100 cents, divided into logarithmic units. And we can create these little melodies. And we ask people to distinguish between them. And they sound like this. If that's using 60 cents, which is 60% of a semitone. You can definitely hear that difference. And we titrate it so that this is a much smaller difference of 10 cents. It's more or less close to threshold, I would say. You can hear some differences there, but much, much smaller. And so we designed a learning paradigm where people were tested over 3 days and given feedback as they performed the task. And subjects were divided into stimulation groups. They either were stimulated over the right, over the left, or had a sham procedure. And we can derive these nice psychometric curves. So for each individual, we have a threshold. And that's what I'm plotting here is the threshold level relative to a baseline. So we baseline correct. Essentially, what you see is that people do learn this task. Their thresholds get lower over the 3 days when they're stimulated on the left or if they have a sham procedure. But if they're stimulated on the right, their threshold actually gets slightly worse, and they certainly don't learn. So we see this behavioral difference according to the side of the stimulation. What's important here is that because we are recording within the MEG scanner, we can actually look at the physiological response and see how it's modulated. And in this particular study, we simply looked at the magnitude of the N1 response. So a very simple evoked response here. And what we see is that indeed in the right-sided group, the amplitude drops after stimulation. And in the left-sided group, the left side amplitude drops. So there's a bilateral effect. Depending where you're stimulating, the ipsilateral auditory cortex gets depressed to some extent. But what's important is that behaviorally, only the right side showed the effect on pitch. And indeed, when we look at the correlation between the change in pitch threshold, in other words, how much you learned, and the change in the N1 amplitude, in other words, how much the stimulation dampened your response, we see this correlation using both during and after measures, whereas we don't see that correlation on the left. So the stimulation does have this bilateral effect, but only the one on the right actually predicts something about the behavior. So this is all consistent with this idea of fine-grained analysis of pitch being more important on the right. Further evidence using a very different paradigm comes from this study done by my former student, Emily Coffey. And what we did here was use, again, magnetoencephalography to look at the frequency following response, a potential which is very commonly used in hearing sciences, typically with EEG, but we did it with MEG because that way we can localize the sources of the response. And as its name implies, the FFR follows the frequency of the audio signal. So it's sort of a low-pass version of the audio signal. When we looked at the location of the response, we see that it's stronger on the right than on the left in the cortex. There are also generators, as is well known, in the inferior colliculus and medial geniculate, which we can see with MEG, but we can also see the cortical response, which hadn't really been recognized until Emily discovered it. It's now been replicated by several other labs. And what was really cool for us is that the response, as you can see, is much larger on the right. Here's the distribution of each individual subject, and you can see that it's extremely skewed right greater than left. And what's also very interesting is that the amplitude of the response on the right side is correlated with frequency discrimination thresholds measured in a traditional psychophysical way, whereas there is no such correlation on the left. So this again links brain to behavior, just like in the direct current stimulation study. So we have a lot of different converging evidence in favor of this idea. Now we're sort of honing in a little bit, going from, well, it's got something to do with pitch. Now we're saying maybe it's got something specifically to do with fine-grained pitch. Having done that, now we have to ask ourselves, well, why might that be the case? What model might explain this? And in particular, we wanted to pursue the idea of a complementarity of specialization between right and left. And so to look at this, we've done a series of different experiments. This one was carried out by Pascal Belin, who is now a professor at Aix-Marseille, and with whom I did a sabbatical just last 2 years ago. And with Pascal, we devised this imaging protocol where we take some tones and we vary either the speed at which they alternate, so going from relatively slower to relatively faster, or we keep the speed constant and vary the size of the frequency intervals. So we divide the octave into halves, quarters, eighths, and so on. And then again, we look in the brain with fMRI. We use the values here as regressors to see where the responses occur. And we do see bilateral responses to each of these parametric changes, speed of alternation and degree of frequency resolution, if you will. But what's interesting is that even though there are two blobs here for the temporal component, we get an overall greater response on the left than on the right. And conversely, we get an overall greater response on the right than on the left for the frequency variation. So a nice double dissociation. This is now a fairly old study from 2001. Several other studies from several other labs have used, I would say, complementary approaches, looking at similar phenomena, but using very different stimuli, using very different procedures. One was done by Mark Schoenwiesner, Bohemio from David Popel's lab, and Overath from Tim Griffith's lab, have all done different kinds of experiments. I won't go through the details. I would just say that all these experiments are consistent with the model that we're thinking about. And that model was sort of articulated in this paper that I wrote with Pascal and Virginia, where we argued that auditory cortices in the two hemispheres are specialized such that temporal resolution is better on the left and spectral resolution is better on the right. Something that's important here is that it's not really necessarily just periodicity, because in some of these experiments, people are using sort of different noise bands or speech-like sounds. You still see the same sorts of relationships. So it need not be specific to periodic stimuli. I think it's probably broader than that. So now we have a more, I think, maybe elegant model where we're saying that there's the difference between left and right has to do with degree of spectral versus temporal resolution. So now we can, now that we've identified that model a little bit better, we can now try to look more at the underlying physiology. So we have two pieces of evidence that support the idea. The first has to do with connectivity profiles. And let me explain what I mean by that. This is from a study done by Kuok Cha, who was a student of mine, and Mark Schoenwiesner. Mark is now a professor at University of Leipzig. And what we did here was, if you just look at the top row here, very simple fMRI experiment presenting different frequencies of tones. And we get these sort of tonotopic maps. This is pretty standard, where you simply identify each voxel according to its magnitude of response, and you assign it to a given frequency, depending on whether it's more responsive to lower or higher frequencies. But then what we can do is we can take any given voxel, as shown by the crosshairs here, and we can ask how, to what extent is that voxel functionally connected to all the other voxels that are active. So we just look at the time series of the response in the fMRI, and we correlate them. And if they're going in the same way, it's a high correlation. If they're not, then it's a low correlation. And then we get this functional connectivity map. And you can see that there are areas of high correlation, surrounding the target voxel, but also quite distant, even in the other hemisphere. And so we do that for every single voxel in the entire map. And that gives us these sorts of functions. So here we're looking at the degree of correlation for a given voxel according to the best frequency of all the other voxels. And the red line indicates the position of the target voxel. So you can see that there's more correlation between the target voxel and all the other ones when they're of a similar frequency. And you see that peak here. If we shuffle these, we get these graphs like this. We're just shuffling it according to delta best frequency. And then we average all of these, and we get these kinds of graphs. So across all voxels, this is telling us how strong is the correlation, how strongly is the correlation affected by the frequency, by the best frequency of that voxel. So we're playing a game as if we are actual physiologists. We're not able to look at single neurons. We're able to look at single voxels. Single voxels contain tens of thousands of neurons. Nevertheless, we still see some of these properties, which is sort of nice. And the reason this was exciting to us is because we found this difference between the connectivity profiles in left core areas and right core areas. In both cases, you see this drop-off in degree of functional connectivity as a function of frequency, but the drop-off is much steeper on the right than on the left. It's much flatter here. And if we quantify that difference, we see much greater slope on the right than on the left. By the way, we saw this with the stimulus-driven epochs. We also had resting epochs in between the stimulation, and we actually see the same thing. So this is sort of an internal replication. We can see it in each individual subject even. These are our individual participants. You can see the same pattern. And there was some interesting neurophysiology data coming from, I believe, the rat in this paper by Liu et al. And they showed that by dampening the intracortical responses using a pharmacological agent, they found that the thalamocortical inputs are not as highly tuned because when you remove them, you get this broadening of tuning here shown in red. And so their conclusion, as you can read there, except I can't read it, that there's a selective amplification of cortical cell responses at preferred frequencies by intracortical inputs from similarly tuned neurons, which is, I think, extremely consistent with what we're finding. So neurons that are tuned together, that are tuned similarly, are interacting more, and this leads to higher frequency selectivity. So we have identified one potential mechanism, which has to do with this sort of local connectivity profile. And the second mechanism has to do with spectrotemporal modulation. Many of you will be familiar with this work by Frédéric Dunissen, who's worked primarily in birdsong. But the idea here is that you take a spectrogram, in this case it's a spectrogram of speech, and we're all familiar with this representation of sound, but you can take this and represent it in spectrotemporal modulation space. And essentially, you can think of this as a filtering procedure, so that if you have, let's say, a sharp onset, as you do here, then that is a moment in time when there is rapid temporal modulation. And so that will be represented here along the temporal modulation axis as being relatively high temporal modulation, but relatively low spectral modulation. And this contrasts with, let's say, a moment here where there are many harmonics, so it's a very rich sound in terms of its harmonic structure. And so in that case, it would correspond to a point here, which has relatively high spectral modulation, but relatively low temporal modulation. And this idea has been formalized by Shihab Sharma, who, as I think all of you know, has done a lot of work here in Paris. And what Shihab proposed back 20 years ago is that these spectrotemporal receptive fields that you can record, not only from cortex, but from many other structures in the auditory pathway, you can relate this to these spectrotemporal modulation patterns by a 2D Fourier transform. So when we represent sound this way, we're representing it in a way that arguably is physiologically meaningful, because neural units are not tuned only to one parameter. They're jointly tuned both to spectral and temporal features. So can we actually investigate these spectrotemporal modulations using functional imaging? This is from an experiment that Mark Schönwieser and I did a few years ago, just to demonstrate that indeed you can. So for this experiment, we use these sort of ripple sounds, where they vary independently in their temporal modulation rate or spectral modulation rate. And they sound a lot like Space Invaders video games from the 1970s. So that's sort of slow temporal modulation and low spectral modulation. Here's a slow temporal modulation, but much richer spectral content. And here's the opposite, which is very fast modulation. And when we do that, we find, we get a map for every given voxel. We get a map of the magnitude of the response using all 8 by 8 stimuli here. It's a bit of an extremely boring experiment to have to listen to these sounds. And so for each voxel, we can identify one of the stimuli which happens to give it the greatest response. And when we look at all the voxels, we see a variety of patterns. Each one of these represents a voxel. And we do see some that respond only to one or a small number. And some of them are sort of, tuned to spectral modulations. So this one prefers low spectral modulation. This one prefers high spectral modulation. But they don't care so much about the temporal. Others are more tuned to temporal modulations and don't care so much about spectral. And many of them are jointly tuned or have multiple areas. Now, when we go back to our obsession with speech and music, if we represent speech and musical sounds in this spectrotemporal modulation space, we notice something interesting, at least I think it's interesting, which is that when we look at musical sounds, they tend to have relatively high spectral modulation, but they're on the left of this graph, so they have relatively low temporal modulation. When we look at speech, we see kind of the opposite. A lot of temporal modulation, but not so much spectral modulation. And this idea that there might be a difference between speech and music was also taken up by folks from David Popel's lab. And here they're just plotting the temporal modulation rates for a large range of different speech sounds from many different languages. And you see here in the black that the average modulation rate is around 6 or so hertz. Whereas for music of many different styles, the average modulation rate is more like 2 or so. So there seems to be a big difference in temporal modulation between the two. So how can we test the meaning of this? This is simply descriptive, right? How can we test the importance of these cues to speech and music? For this, we go back actually to Frederick Tunissen's work, and we borrowed the idea that once you've represented the sound in this spectrotemporal modulation space, you can selectively remove some of the information either in temporal or spectral dimensions. And that is exactly what we did with my former student Philippe Albouy, who is one of the few Frenchmen we've been able to retain in Canada. He's got a position now at Université Laval. And Benjamin Morillon, who is a colleague also in Marseille. And what we did here was we took a sound, which I'll play for you in a moment. We put it into this spectrotemporal space. Then we apply the filtering using algorithms similar to those designed by Tunison. And then we reconstruct, just for purposes of showing you what it looks like, we reconstruct the spectrogram. And so in this case, for example, we are removing almost all the spectral information, leaving the temporal information intact. And here we're doing the opposite. We're removing the temporal information and leaving the spectral information intact. The sounds that we used, we went to a lot of trouble to hire, first of all, hire a composer. Then we hired a soprano, and we recorded these wonderful songs that you can see here. And the stipulation was that they all had to have the same rhythm. However, the tonal patterns are all quite different. They're all in different keys. And the speech content is different. So these are sung sentences, okay? And of course, because we're in Montreal, we had our French version and we had our English version. Because we had 10 different sentences and 10 different melodies, we could create this matrix of 100 songs where every sentence appeared once paired with every melody. So we have 100 unique songs, but we can focus only on the melody or we can focus only on the speech. And so this is what happens to these songs when you put them through the different filters. And I'm sorry, this is going to be very, very soft here, but let's see if you can hear it. So this is the original song. I don't know if you can hear that. We tried everything to make it louder. We couldn't do it. But this is the song before any distortion has been applied. Now we're going to degrade spectrally, remove some of the spectral information. Je voudrais parler à mon bon copain. You can still understand that, right? Je voudrais parler à mon bon copain. And here is when you remove almost all the spectral modulation. Je voudrais parler à mon bon copain. So it's sort of Darth Vader speech, but still perfectly comprehensible. But the melody is completely gone, right? When we do the opposite, now we're going to do temporal filtering. We are going to take the original melody. This is one of the English examples. The people thought a coin was hidden there. Now we're going to do some temporal degradation. You can maybe get some of the speech there, but by the time you do a lot of temporal filtering, I'm sorry it's so soft, but you can get the idea, which is that we're basically removing all these temporal cues. That's why it's as if we're brushing the spectrogram, right, with a brush and increasing the correlation across time. And so by the point you get here, you have this sort of fuzzy thing. When we ask people, let me just skip through this for the interest of time. So when we ask people to perform a simple sort of match the sample task, we ask people they always hear the song, which contains both a melody and speech, but we ask them either to focus on one or the other. When we do spectral degradation, and we ask them to focus on the melodies, the more we do the degradation, the worse they get. So the task becomes nearly impossible. it's nearly impossible to perceive the melodies when you degrade the spectral information. But the same stimuli, when you ask people to identify the sentences, there's absolutely zero change. That's the Darth Vader talking that I showed you. So this is a very noticeable difference. And when we do the opposite, temporal degradation, we get the exact opposite pattern. Temporal degradation does nothing at all to melody recognition, but strongly degrades speech perception. So this is a very nice double dissociation here. This was for the French sentences. The exact same pattern appears in a separate group of individuals who were Anglophone speakers who got the English version of the stimuli. So perfect replication of the behavioral results. And very recently, with my former student Yi Du, who's now a professor at the Chinese Academy of Science in Beijing, she created some Mandarin versions of these sentences. And we just got these results a couple of weeks ago. It's very exciting because even though Mandarin is a tonal language, when you degrade the spectral information, it does absolutely nothing to the comprehension of the Mandarin, but it does degrade the perception of melodies. And vice versa, the perception of sentences is degraded with temporal with temporal filtering is applied. So replication even in Mandarin. Now with Philippe and Benjamin, we of course looked at the functional MRI response, and we used a classifier. This is a machine learning multivariate analysis where we look to see how well can the decoder determine if which stimulus is which. Now remember, we had 10 different sentences and 10 different melodies. So this is a 10-way classifier, which is pretty hard for classifiers to do that. Usually it's done with like 2 categories. So we're just plotting the accuracy, which is chances 10%, of course. And what we see is that when we ask the classifier to identify which sentence is which, we get above chance classification from this left auditory cortical region, but chance performance on the right. And vice versa, when we ask the classifier to identify which melody is which, we get significant classification on the right, but not on the left. And when I saw this, I thought way back to the early 1990s, well, this looks kind of familiar, except that then we had no idea what was going on. We didn't have a model, we didn't have an explanation, we didn't have a kind of understanding of what was behind it. And I think now, you know, it's not the empirical data, it's the understanding that the empirical data allows you. And I think we've come a long way now to be able to figure out what's going on. In the same experiment, we of course look at the degree to which the classifier is affected by the degradation. We know behaviorally what happened, I just showed you that. What happens to the classifier? Well, let's look at the left auditory cortex first. When we ask the left auditory cortex to classify the sentences, shown in blue, performance is very good under spectral degradation, but performance drops to chance under temporal degradation. This is the classifier performance, right? Not the behavior. This is what's going on in the brain in that location. For the melodies, the left side doesn't do very well at decoding melodies anyway, so it's really not different from chance. On the right side, we have exactly the opposite pattern. Temporal degradation doesn't affect the classification of melodies from the right auditory cortex, but spectral degradation drops it to chance. And the right auditory cortex is not good at sentences anyway, so it doesn't change much. So this is, I think, the first time I've ever done an experiment with a triple dissociation, which is really pretty cool. And one last thing we wanted to do was to look at the pattern of errors, because I've shown you data from the classifier, which is coming from the brains of the participants, and the behavior, which presumably is also coming from the brain of the participants, right? So there ought to be some relationship between the errors that are made by the humans when they're responding and by the brains of those humans when we're measuring their brain activity. So basically we derive a confusion matrix with the actual correct performance and the predicted performance. We remove the diagonal because we already looked at the overall percent correct. We just want to look at the errors. And then we do this procedure, which is this normalized mutual information, which don't ask me about it because it's Philippe who's the expert on it. But it's basically a way of looking at the correlation in the pattern of errors in this confusion matrix between the behavior for each individual and for each voxel of the brains of each of those individuals. And to make a long story short, we get the pattern that we expect, which is that there's a greater correlation on the left side in terms of the pattern of errors for the speech and vice versa for the melodies. So, sorry. So just to kind of conclude and bring it all together, I've shown you evidence that there is this specialization of left and right auditory cortices for the either integration of spectral information and resolution of temporal information, that's the left side, or segregation of spectral information and temporal averaging on the right side. We believe that this can be explained, in part at least, based on these local connectivity profiles, which would seem to be related, because one thing I didn't mention is that on the left side, you have less sensitivity to frequency, but another way of thinking about that is greater integration across frequency. And there's reason to believe why that greater integration would also lead to faster throughput. And finally, I just showed you this idea that there's this differential effect of the filtering, which means that music and speech tend to exploit different ranges of the spectrotemporal modulation space. So it's not that, like we thought in the old days, that speech is on the left and music is on the right. That, I think, is much too simplistic. What we think is that music and speech tend to use spectral and temporal cues differently. And so that's what leads to the differential hemispheric response. Or another way to think about it is that the specialization of the two hemispheres provides a solution to the computational problem posed by the acoustical environment, which is that the acoustical environment has spectral and temporal modulations, and you can specialize for one while leaving the other one aside, shall we say, and vice versa. And I just want to finish, if I have like 2 more minutes. Am I okay, David?
Another 7 minutes.
Okay, thank you. So, normally I would just end here, but I thought important to give you a kind of a broader perspective about, sort of why should we care about this? And in particular, why should we care about music and why is it important? And so, why do we have these two communicative systems? Why isn't speech enough? We can say everything we want to say with speech, right? And I think there's, the reason is it has to do with what sort of Darwin proposed in a sense, which is that music is related to emotional arousal, to the expression of emotion, to the regulation of emotions, both self-regulation, but also the influencing the emotions of others. And we could go so far as to say that it promotes psychological well-being. And I think that a lot of this has to do with the fact that music engages the reward system. And this is, an area of research that I've done a lot of work in. It's not really related to the work that we're discussing today in terms of the prize, but we have done a lot of different experiments using different protocols to look at pleasurable music. And over the years, we've seen very consistent responses in the striatum, which is part of the reward system, probably a core region of the dopaminergic mesolimbic system. And we see this very consistently. And in fact, recently we published a meta-analysis with Ernest Macerrero, one of my former postdocs, where we see across a large number of studies, we see responses in dorsal and ventral components of the striatum, as well as the ventromedial prefrontal cortex, which is another key component of the reward system, and of course, the superior temporal gyrus. So we have both the auditory component and the reward component. And indeed, we believe that it's the interaction between these two systems that enables us to enjoy music, to derive pleasure from it. And we have lots of different evidence for that. I'll show you just one piece of evidence from another study we did with a group in Barcelona. If we look at the functional connectivity between auditory areas and the ventral striatum, what we see is that the connectivity increases as a function of the degree of hedonic response that the participants express. So if we divided our groups into those who really have zero hedonic response, we call them anhedonics, there's basically no connectivity between those two systems. Whereas the average listener has an average level of connectivity, and these are like music lovers who have an even higher degree of connectivity. So we really think that the experience of pleasure is associated with this interaction between these two systems, the reward system that I haven't talked about in this lecture, and the auditory system, which I have talked about. So, in other words, in order to gain access to the reward system, in order to gain the psychological well-being and the hedonic and affective responses that are associated with music, you need to have a fully functional auditory system, because that's the input, which then feeds back. So if I just go back to the very beginning, the fact that we have these two systems, I think in the context of the magnificent work of this institute, I would like to suggest the idea that if we're going to give people the full experience of human auditory communication, we actually need to give them both speech and music. And in particular, based on the research that I've shown you, we need to consider both the importance of music, but also the differential nature of the perceptual analysis that's required for music, which differs from that which is required for speech. And with that, I will close and be sure that I thank all of my many colleagues over the years who've contributed so much to this work, in particular Philippe and Benjamin, who did this wonderful study I showed you at the end, and Pascal, with whom we continue to work on lots of projects, and my closest collaborator, Virginia Penhune, and of course all of the granting agencies. And I'm very, very proud to now be able to add this logo to every one of my slides from now on. Thank you very much.
I think we'll proceed without the technical challenge. I think I'm going to come and join you over here, Robert. So thank you so much for what was an absolute tour de force of methodology, as well as, I think, extreme interest in neural behavior, and different aspects of neural function, and all addressing questions that are of fundamental importance to what makes us distinctively humans, as I think was shown in the introductory slide. And we have plenty of time now for questions. I've got a whole sheet of them over there that I've been taking during the talk, but can we start with Larry, please?
Well, I'll echo that, and thank you for an amazing and thought-provoking lecture. I was particularly struck when you were playing your simulated recordings where you changed spectral and temporal relationships. And when you dropped out the spectral information, it sounds surprisingly like all of the simulations that you give for cochlear implant users.
Indeed.
And as we know with cochlear implant patients, we give them really good temporal information, and they all tell us that music sounds horrific. So I'm wondering if you could comment on what it is we're doing wrong with cochlear implants to not allow them to be able to hear music. Is it the way we're coding it? Is it the way we're stimulating the neurons? Or is it the fundamental biology of the sickened ear that we just can't create that? What are your thoughts on that? I'm sure you know a lot about that.
Thank you very much. That's a great question. And I should note for the record that Bob Shannon showed this effect on speech. You may recall he had a science paper, 1981, I think, where he showed, he was putting a positive light on it, saying that speech perception is possible with a small number of spectral channels, which in a way was, part of what precipitated the idea that with a limited number of inputs to the cochlea, you might still be able to perceive speech. And you're absolutely right that what I refer to as Darth Vader speech is similar to some of those algorithms that are supposed to simulate what a person with cochlear implant is hearing. Now, I'm far from an expert on cochlear implants, and many of you are, and so I'd be very happy to continue this conversation with those of you who have the expertise. But from what I understand, the main limitation is simply physical in terms of the number of input channels. So I don't know what the latest generation of implants are like. How many electrodes can you actually put into the cochlea?
Well, you can put in 22, 24, but we don't even think patients are even using that many.
Because they
current spread
and they interfere with each other. Yeah. And so, what's the normal number of hair cells is like 30,000 or something like that, right? So you're going, several orders of magnitude down in spectral resolution. How to fix that, I don't know, but I think it is something, as I said sort of towards the end there, maybe a bit provocatively, that it's something that it seems to me would be very important to try to work on. I don't really know how one might do that. We did do a little conference a couple of years ago with my colleague Alexandre Lehmann, who's very interested in this idea. And we had some of the industry people there, and we had testimonials from musicians with cochlear implants. There are several interesting things. There's a lot of individual differences. There are some people, few, but some, who say that they do enjoy music. They tend to be people who are postlingually deaf, so they had already existing representations, and maybe the degraded input is still enough to trigger that. Another intriguing idea is that I've emphasized the spectral aspect of music, but there's, of course, very strong temporal cues in certain styles of music. So if you're only looking at drumming, for example, it's primarily temporal. And so there's the idea that maybe if you focus more on percussive sort of music, that might work. There was even the proposal that was floated at this meeting of getting composers and performers interested in generating music that is sort of engineered to have more temporal information. And that could be interesting. There's the interesting idea of maybe promoting rehabilitation approaches. Actually, something Virginia works on is the interaction between auditory and motor systems. And so perhaps by engaging motor systems during the rehabilitation, you could get a better fix on the temporal structure of the music. So there are a number of ideas floating out there, but I think it's an unsolved problem, and I for one would not want to get an implant for that reason. I'd rather keep my hearing.
Yeah.
Hi, thanks. Congratulations and thanks for the great talk. My question is regarding the first part, the difference of hemispheres, left and right. How that is conserved in other species, for instance, in birds, singing birds versus other birds?
Yeah, that's a very interesting and complicated question. There is, in fact, some evidence of lateralization in some bird species. It's a little tricky to do the comparison because the bird brain is in evolution so far from the human brain. The evidence from closer species, like chimps and macaques, is very mixed. It's not nearly so clear as it is in a human. You will find studies that do report some hemispheric differences. You find lots of others that don't. One of the big limitations, and I've spoken with neurophysiologists about this, is that it's a practical limitation. When you do neurophysiology, you only put the electrodes in one side, right? So it would be, I think, complicated, not impossible, but quite complicated to put electrodes on both sides and do the comparison. I don't actually know of any studies that have done that. You can, of course, now do studies with primates in functional scanner. Actually, Pascal Bellin is doing some of that in Marseille. We went to visit his lab and saw the monkeys, being placed into the scanner. It's very cool. So that might be a way to address it. But I would say the evidence is weak for any clear-cut specialization. And maybe that's because most communicative systems in most species are is just one sort of system, right? I try to emphasize this idea that we have two parallel systems, and that's not generally the case. If a bird has a bird song, it may have several different songs, but it doesn't have an entirely different class of sounds that it creates for some other purpose. The same with monkey vocalizations, right? So
maybe second related questions, but more clinical, and maybe Larry, you may comment. Because of these differences, I may have read that when in clinics, when we implant, and we have just one cochlear implant, which is for hearing, so we implant in the right ear so that he can benefit from the left hemisphere. So my question is, are there differences for patients who are unilaterally implanted? Difference in performances for one implanted in the right versus the ones implanted in the left ear?
that's a really good question. I don't know the answer to that. I mean, we know that the auditory pathways, of course, are bilateral. So if you implant one ear, it's still getting to both hemispheres. But we also know that the contralateral projections are stronger. So I don't know that literature. Maybe somebody in the audience does. That's a very interesting question.
Well, that's exactly what we do.
Can we have the microphone, please?
So that's exactly what we do. Anybody with any residual hearing on one side, we always implant the worst ear. And what patients will tell you is that they do better with one ear in some situations, like listening to music, the non-implanted ear. They do better with the other ear in just one-on-one listening conversations. But together, they do much better overall. And so, but it takes about 6 months to a year for that to happen. The brain's got to figure out how to integrate these wildly different signals. And the older you are, the longer that takes.
But that's an important point. The complication, to address your question, is that you will have the input from one ear, which is going through the implant, but you still have some input from the other ear, typically. And so it's kind of a mixed input, right? I mean, maybe that's something that could be studied in people who really have bilateral complete deafness. That it might be a cleaner population to study.
Thanks a lot for the crystal clear talk. So I was wondering, so music perception is not only done of what you showed, so fine grain frequency discrimination, but also of octave equivalence, and even more like temporal capabilities, like a tempo or rhythm perception.
Yes.
So have you looked to this other capability, and whether there is kind of bias also between left and right?
Yeah. So obviously, this is why I sort of said it's not so much that music is on the right and speech is on the left. It's more about the type of cues. So if you have a musical event that has rapid temporal changes, my prediction based on the model would be that, in fact, it would be better processed by the left auditory cortical pathways. That said, as I showed you on that graph from David Popel's group, the average temporal modulation rate of music is just slower So music tends not to have those features, but it could have those features, and in some cases it does. Now, in the case of higher order temporal representations like metrical structure, those are typically quite slow, right? So if you could have a lot of fast events, but it's still going to be there are hierarchical levels of meter, and the higher you go, the slower they are. So you could represent temporal information integrated with the sort of pitch information better on the right, as long as it's slow, because the right side is fine as long as things are evolving slowly.
Thank you for the really great talk. It was really fascinating to see this dissociation of spectral and temporal information between left and right. But in the end, the percept that you have is fusing the two information. So do you think you need to fuse the information from the right and left cortex? And where would this happen if this is needed?
Yeah, that's a really excellent question, and it's one that I've been thinking about lately, because our emphasis has been on trying to, dissociate the underlying mechanisms to see if we really can demonstrate that they're separate. But you're absolutely right. The percept is an integrated one, and particularly in the case of song, which is what we were using, you do have integration, and we know that from psychological experiments that actually Sabrina and I did one, I think in 1987 or something, a long time ago. And there's evidence that the speech content and the musical content are actually integrated in memory, so that when you remember something that you heard the previous day, the words and the melody sort of form a single unit. So how that happens, I think, is a super interesting question for future research. Now that I think we feel pretty comfortable with the idea that there is this segregation of function, we have to understand how it comes together. My guess would be that there's a lot of crosstalk between the auditory systems and the two sides. We know that there's a huge pathway that goes through the corpus callosum that connects the two auditory systems to one another. That hasn't really been studied much in this sort of context. It's been studied, I think, in the context of binaural hearing and spatial processing. But in the context of something like music versus speech, it hasn't really been studied. So I think that's a very interesting area for somebody to write a grant about.
Thank you for this very exciting talk. I have a question about the part of spectral degradation and temporal degradation too. You have shown individual data. It seems that there is a huge variability among subjects, or more precisely that some subjects act very differently compared to the average of subjects. It's no, the one? No, yes. Yeah. No, yes, it's this one.
Yes.
How do you explain that?
Yeah.
Do you have any explanations in terms maybe of age, gender, or for something else?
Yeah, thank you. That's a very interesting question. It's the reason why we put all these lines here, is to be sure that we show, the actual data, rather than just rely on the averages. I think what you see is much less variance in terms of the effect of spectral degradation on speech. The blue lines here are almost all flat. But you see a much greater variance in the processing of melodies. There are some people really who aren't affected by the spectral degradation. Most people are, but some aren't. We see that in both groups. So I think this is a curiosity. Why is that the case? Some of it I would attribute simply to noise. The melody task overall is harder, so we've normalized here. But we tested people who were not musical experts in any way, so they didn't have any special musical training. And so that's one issue, because none of the participants had musical training, but all of the participants were expert speakers of their language, right? So that might account for part of it, just the familiarity and the cognitive capacity to process melodies might be not so developed in the average listener. I think that accounts for part of the variability. Why do some people, why are some people not as affected by the spectral degradation? We have not found any clear reason in terms of individual demographic variables. I think it would be interesting to follow up on that, to find people who consistently show or do not show an effect, and then, for example, put them in the fMRI scanner and see if we can understand why that might be the case.
I take the privilege of the last question, Robert, and ask, it seems in the top left there that those who are not affected by it are mainly those who score on the top end of the spectrum. So the normalized score there, yeah.
Yeah, well, but there are some down here.
So, yeah, but they seem to be biased towards the better performing individuals.
Yeah, I mean, I think that's a good point. So it may be the case that some people are more resistant to the degradation, and it's also a matter of how far you take the degradation. If you keep going, you can degrade, all the way until you have no modulation left, at which point there will be no information being carried in terms of spectral modulations, at which point performance has to drop to zero, right? Because it'll just be, it'll essentially be like an amplitude modulated noise at that point, right? So that would be another answer to the question is, well, what if we expand the range here? Will these people eventually fall down? They have to, but it might be more informative to think about it in terms of the shape of their function as opposed to the absolute level of performance. And so it could be that different people have more or less resistance to the spectral degradation. I personally would like also to see how that varies with musical training, because you could sort of make two opposite predictions. You could say, well, musicians are trained to spend a lot of time listening to spectral information, so maybe they're more dependent on it. Or you could think, well, musicians, by virtue of having a lot of training, maybe have a greater capacity to represent the information, even with little bits of it, and so they might be more resistant. And so I'm not sure which way it would turn out, but that's an experiment that will go into the next math application.
That seems the perfect time to wind up the present presentation, and I think we could happily go on discussing lots of implications of these data all day. But could you please congratulate Robert and thank him for his talk once more?